Inside every single one of your cells, right now, there are these tiny microscopic machines just buzzing with activity. They're called proteins, and they do, well, pretty much everything. But here's the crazy part. How does a simple string of chemicals fold itself into the perfect 3D shape to do its job? That's the amazing puzzle we're going to dive into today.
So, this is the big question, right? How does a one-dimensional chain of chemicals, just a line, know how to twist and bend and fold itself into a complex, working, three-dimensional machine? It's an incredible journey from a simple line to a complex shape, and honestly, it's one of the most elegant processes in all of biology.
So, here's how we're going to break it down. First, we'll look at the blueprint, the basic instructions. Then, we'll see how the very first, simple folds start to take shape. After that, we'll watch the whole thing collapse into its final 3D form. And finally, we'll connect that exact shape to what it actually does.
If you take away just one thing today, make it this. For a protein, its structure is its function. The shape is everything. Think of it like a key for a lock. If the key has the wrong shape, it's useless, right? Same thing here. The entire folding process is all about reaching one very specific functional shape.
Okay, so everything has to start somewhere. And for a protein, that starting point is the most fundamental level, what we call the primary structure. This is the protein's initial set of instructions.
This is where it all kicks off. It's just a simple chain of building blocks called amino acids. And this chain, this is the protein's primary structure. The best way to think about it is like letters spelling out a word. The specific sequence, which amino acid comes after which, is absolutely critical. It dictates all the folding that's about to happen.
A great way to picture this is with Lego bricks. Imagine you have about 20 different kinds of Lego bricks, all different shapes and colors. The primary structure is basically the instruction manual that tells you, okay, put a red brick here, then a blue one, then a yellow one. And that instruction manual, it comes directly from our DNA.
Now, the bonds that hold these amino acid bricks together are called peptide bonds. And they have one really crucial quirk. They're rigid. They're flat. They can't just spin around freely. This might seem like a small detail, but it's actually the very first rule of folding. It means this whole process isn't random. The protein is already limited in how it can bend, right from the very beginning.
All right, so we've got our chain of Legos, and the connections are a bit stiff. Let's move to the next level. That linear chain now starts to form these local repeating patterns. This is what we call the secondary structure.
The thing is, just being a long, floppy chain, it's not very stable, it's kind of messy. So, to become stable and actually do something, it has to start organizing itself into these neat local patterns.
And it turns out there are two super common patterns that pop up all the time. On the left, you've got the α-helix, which looks just like a spring or an old-school telephone cord. And on the right, you have the beta-sheet, where the chain kind of zigzags back and forth on itself, forming this flat, pleated sheet.
Let's zoom in on that alpha helix for a second. See how it's this really stable right-handed coil? And notice how parts of the amino acids are sticking outwards from the center? Those are what make each amino acid unique. And trust me, they're going to be really important in just a moment.
So what's holding this springy shape together? Tiny little forces called hydrogen bonds. Now, individually, they're pretty weak, but there are a ton of them. In this helix, they form between amino acids that are just a few spots away from each other in the chain, acting like tiny staples that lock the whole coil in place and make it super stable.
Okay, we've got our blueprint, the primary structure, and we've got our first local folds, the secondary structure. Now, it's time for the main event, seeing how all these helices and sheets fold up together to create the final, functional 3D shape. This is the tertiary structure.
The process really happens in stages. First, those local alpha helices and beta sheets just pop into existence along the chain. Then, these different sections start to fold and pack against each other until the whole thing collapses into this specific, compact blob. Let's actually watch that happen.
So here we go, we're looking at one part of the long chain, and boom, a local fold, in this case an alpha helix, has just formed. That's our starting point for the big fold.
And now, let's zoom out. You can see how multiple alpha helices, all within that same single chain, have now folded together. This protein here is myoglobin, and you can see it's creating a much more complex arrangement. The tertiary structure is really taking shape. And there it is, the final result. This is the fully folded protein. It's not a string or a few coils anymore, is it? It's a dense, compact machine with a very specific surface, all ready to get to work. The inside is just packed together, maximizing all those forces that hold it stable.
But what's the engine driving this big collapse? Well, for the most part, it's something called the hydrophobic effect. See, our cells are mostly water, and some parts of the protein chain are water-fearing. They hate water, so what do? They all run and hide together, burying themselves in the core of the protein. At the same time, the water-loving parts are happy to stay on the outside. This is probably the single biggest force that locks in that final shape.
So we've seen how it folds. Now let's talk about why it matters by putting that core principle, structure determines function, into action. We'll look at two major types of proteins and see how their final shape is perfectly suited for their job.
When all is said and done, the final 3D shape usually falls into one of two main camps. On one side, you have fibrous proteins. Think long, rope-like structures. They're built for strength and support. Then you have globular proteins. These are compact, spherical little machines, and they're designed for action. Things like carrying molecules or making chemical reactions happen faster.
And wow, this slide just says it all, doesn't it? On the left, that's alpha-keratin, the protein that makes up your hair. It's basically a bunch of alpha-helices coiled together into a super strong filament. Its shape is perfect for providing structural strength. And on the right, there's our friend myoglobin again. Its compact globular shape creates this perfect little pocket to grab onto an oxygen molecule. Two totally different shapes for two totally different jobs.
So let's do a quick recap of the whole journey. It all starts with a primary structure, that specific sequence of amino acids. That sequence then determines the local secondary structures, like the alpha-helices. Those pieces then fold up into the final tertiary structure, driven by forces like the hydrophobic effect. And that final, precise 3D shape is what gives the protein its specific job.
And all of this leaves us with this incredible, kind of mind-blowing question for the future. We're getting better and better at predicting how a protein will fold just by looking at its sequence. So if we truly master these rules, what could we build? Could we design brand new proteins to fight diseases like cancer, or create enzymes that eat plastic waste? The possibilities, they're just extraordinary.